When it comes to fighting deadly infections, the real battlefield is totally invisible. It's inside our own bloodstream. So today, we're going to look at the incredible tech that acts like a frontline scout, finding these hidden enemies before things get really bad. And that really is the million-dollar question, right? When you're dealing with something like sepsis, every single minute, every minute counts. The real challenge isn't just finding the bug, it's finding it fast enough to actually do something about it. So how on earth do we win that race? Well, the answer is surprisingly cool.
It turns out the secret isn't to look for the microbe itself. Instead, we listen for a telltale sign. You can almost think of it like we're detecting the microbe's breath. Yeah, you heard that right. We're not trying to spot the bug, but the evidence it leaves behind just by, being alive.
And what's really fascinating here is that on a super basic level, these tiny microorganisms, the bacteria and fungi we're trying to find, they act a lot like we do. They need to eat to survive. And this whole process, it's called microbial metabolism, and it's the absolute key. They take in nutrients from their environment, and just like us, they release byproducts. And that byproduct, that waste product, that is exactly what we're looking for. Here's how it works. You take a blood sample and put it in a special culture bottle that's packed with nutrients. If there are any microbes in there, they're going to start growing and multiplying like crazy. And as they do that, they release carbon dioxide, CO2. So get this, the crucial part is we don't even need to see the microbe. We just have to detect the CO2 it's breathing out. A rising level of CO2 is basically our smoke signal. It tells us there's a fire. Okay, so the signal is CO2. Simple enough, right? But how do modern labs actually detect this invisible gas? And not just in one sample, but automatically across hundreds of them at the same time. Well, it turns out there are two main ways to do it. Two technologies that are really leading the charge here. The first approach is fluorescence-based. Basically, the machine is looking for a glow, a light that gets brighter. The second method is called colorimetric, and just like it sounds, it's all about detecting a change in color. Both are just these really clever ways of making an invisible signal totally visible. All right, let's dig into that first one. The glowing method. So how in the world does a simple glow tell us that there's a dangerous infection lurking in a blood sample? It's a pretty slick three-step process. So at the bottom of every single culture bottle, there's this special chemical sensor. First, an LED light zaps the sensor. Now, if there's CO2 in there from the microbes, that CO2 reacts with the sensor and makes it fluoresce, or, glow. And here's the cool part. The more CO2 there is, the brighter the glow gets. Then, a detector inside the machine measures that light, and if it gets bright enough, boom, it sounds the alarm. It's basically like a glowing tripwire for any kind of microbial growth. Okay, so that's the glowing method. Now let's talk about the second approach. It uses the exact same principle, detecting CO2, but with a totally different visual cue. We're not looking for a subtle glow this time. No, this method is all about a dramatic, you-can't-miss-it change of color. And this really just nails the concept. The source material puts it perfectly. The sensor literally changes color from blue-green to yellow. You can't get much clearer than that, right? There's zero ambiguity. It is a loud and clear visual sign that something is growing in that sample. So you might be wondering, how does that actually happen? Well, as those microbes release CO2, the gas actually changes the acidity, the pH level of the sensor. This drop in pH is what triggers a permanent, irreversible color change. You can think of it like a chemical traffic light. It starts out, say, blue-green, and as soon as enough CO2 builds up, it flips to yellow, and it stays yellow. The machine spots that color change and instantly flags it for the lab technician.
So, okay, we've got these two really clever ways of detecting infections, one that glows and one that changes color. But the real magic, the true power behind this, is automation. We're not talking about a scientist in a lab coat checking one bottle at a time. No way. This is about a system that literally never sleeps.
And get this. This is the number that changes everything. These automated systems check every single bottle, and a machine can hold hundreds of them. Every 10 minutes. That's 24 hours a day, 7 days a week, 365 days a year. It's a level of constant watchfulness that, let's be honest, no human could ever possibly match.
So, what's the payoff for all this constant monitoring? Well, first off, speed. We're talking about slashing diagnosis times from days to just hours, and that can literally be the difference between life and death. Then there's the automation itself. It massively reduces human error and frees up skilled lab professionals to focus on the really tricky stuff. These systems are also super versatile.
They can spot a huge range of different bacteria and fungi, all using the same tech. Plus, they use smart algorithms to analyze the growth patterns, which makes them even more accurate. But maybe the biggest win of all is that the test is non-destructive. What that means is, as soon as a sample flags positive, doctors can take that exact same bottle and use it for more tests to figure out exactly what the bug is and, most importantly, which antibiotic is going to knock it out.
And all of this really brings us to one final big idea. We've figured out how to listen to the invisible breath of a microbe and turn it into a life-saving signal. It really makes you wonder, doesn't it? What other hidden signals are our bodies giving off? Just waiting for us to invent a technology smart enough to see them and to understand the stories they're trying to tell us.